The physiology of the urinary system revolves around the physiology of the nephron, given that the nephron is the functional unit of the kidney, and the kidney is the main organ in the urinary system. So what does the nephron do? Well, three things. The renal corpuscle accomplishes one of the three tasks. It filters the blood to remove the urea. It is the renal tubule which accomplishes the second two functions of secretion and reabsorption. So the first thing to focus on is the filtration, which occurs at the renal corpuscle. The filtration is similar to the filtration which occurs at a regular capillary that we studied in the chapter on blood vessels. In that, in a typical capillary, there are two opposing pressures. There is the outward pushing blood pressure, and then the resisting osmotic pressure. The same thing occurs at the renal corpuscle with a couple modifications. First, we refer to the outward pushing force as the glomerular blood hydrostatic pressure, the GBHP, as opposed to the regular old BHP. The reason is that, one, since the efferent arteriole, the exiting arteriole, is narrower than the afferent arteriole, that alone will increase the pressure. And then secondly, because there are hormones which allow the pressure here to be slightly different from the pressure elsewhere. So the glomerular blood hydrostatic pressure can be different from regular blood hydrostatic pressure. That is the outward pushing force, which is pushing filtrate through the fenestrations. But there are resisting forces, not only the blood colloid osmotic pressure, because this water here is attracted to colloids, to solutes, and because blood proteins are too big to leave the capillary, there's going to be more solutes in the capillary, so there's an osmotic pressure pushing inwards. But then also a capsular hydrostatic pressure. If there is already fluid in this capsule, that would resist more fluid leaving the capillaries to join it. And so there is one outward pushing force, two resisting forces. When the outward pushing glomerular blood hydrostatic pressure exceeds the other two, then blood can be filtered, filtrate is made, and then urine can be made. The obvious, question is this needs to be regulated, and so if too much blood is filtered and too much urine is made, then there will be glucose and amino acids and salts, which one flushes down the toilet and eliminates as waste. So that's bad. You don't want to lose molecules you meant to keep inside you. But in the same way, you don't want to filter too little. Otherwise, urea, which is toxic, is staying in the body. So because you don't want filtration to be too great or too little, it must be regulated. And it is regulated then at three levels. First, there is what's known as autoregulation, in that the nephron will regulate itself. So let's start off with normal conditions. Under normal conditions, there's a normal amount of blood in this afferent arteriole. That produces a normal amount of pressure in the glomerular capillaries. This makes a normal amount of filtrate that then ultimately becomes urine. And then here there is a normal amount of potential urine passing through the DCT, which is being measured by the maculadenza here. So that is under normal conditions. But under another scenario, let's imagine, look at all that blood. Let's imagine there's too much blood coming in the afferent arteriole. That would create too much pressure in the glomerular capillaries. That would cause too much filtrate, and then that would cause too much urine to be made. These cells of the maculadenza, they are sensing how much salt, and thus how much fluid is leaving here, becoming urine. And if they sense that there is too much, an excess, then they release what's called vasoconstrictor. This vasoconstrictor will then cause the vasoconstriction of the smooth muscle of this afferent arteriole. Now there is less fluid less blood, which comes in the afferent arteriole, less pressure in the glomerular capillaries, less filtrate, and thus less urine. We go back to normal. So the nephron regulated itself. It was the change in the amount of vasoconstrictor made by the macula densa, which regulated the glomerular filtration rate, the GFR. Let's imagine another scenario where now there is too little blood flow here. That causes too little pressure, too little filtrate, and then too little urine is made. That's not good because now toxic urea is not being released from the body. When the macula densa senses that too little urine is being made, it will then decrease the amount of vasoconstrictor made. Notice the green stars are about to stop. With less vasoconstrictor, that then causes vasodilation. With vasodilation, now more blood comes in the afferent arteriole, resulting in more pressure, more filtrate being made, and then more urine. And so once again, it is the autoregulation by the amount of vasoconstrictor released from the macula densa, which is determining this. Now, there is then a follow-up involving hormones, because if there isn't enough blood pressure to create urine, then the obvious question is why? Well, there may not be enough blood pressure in general. And so when there is not enough blood pressure to make urine, the macula densa signals the afferent arteriole, and modified smooth muscle cells in the afferent arteriole, known as granular cells or JG cells, release the hormone renin. Renin then increases vasoconstriction throughout the body. This increases blood pressure. It reaches the hypothalamus, which increases thirst. Maybe the reason that you don't have enough blood for sufficient blood pressure is because you're dehydrated. But then renin is, kind of a boss hormone, which has two underlings. ADH, made from the hypothalamus and released from the posterior pituitary, and aldosterone, released from the cortex of the adrenal glands. And so renin then causes ADH to be released, which will conserve water. And then aldosterone is released, which conserves salt, which then helps conserve more water. And then if the reason that you were not making enough urine was that you were dehydrated, because you're thirsty, now you're more motivated to seek water out. And because ADH and aldosterone allow you to concentrate urine, you will then lose less water. So this now shows that hormones can also then regulate the glomerular filtration rate. As kind of a review, here are two depictions of fluid. There is not only fluid plasma in the vessels, but then here's a kidney tubule, and there is fluid there. And the important thing is to have this fluid in the proper balance. When you're dehydrated, you want fluid to move from this direction, to be reabsorbed from the kidney tubule and to end up back in the blood plasma. Renin causes angiotensin to be released, but then renin also then causes the release of ADH and aldosterone, and these help to conserve fluid. More fluid moves from kidney tubules to the blood plasma, and thus we concentrate our urine. In contrast, if we drink too much water, then the heart measures that there's too much pressure because there's too much blood. The heart then releases a hormone known as ANP, and ANP will cause more fluid to be released from kidney tubules, which will decrease the amount in blood plasma. In addition to autoregulation and these hormones, the autonomic nervous system also helps to regulate the amount of urine made. Under stress situations, if you're fighting for your life or running, then your fluid should be going towards your muscles, not your kidneys. Remember that at rest, at least a fifth and maybe a quarter of the blood leaving your left ventricle can go to the kidneys to be filtered. If there's an emergency, that's simply too much blood. That blood is needed elsewhere. So the sympathetic division will tend to divert blood away from the kidneys. It is only at rest with the parasympathetic stimulation that maximal amounts of blood are sent to the kidneys so that urea can be filtered out. And thus then the glomerular filtration rate, the amount of filtrate and urine made, is regulated three ways: through autoregulation, as the maculadenza changes the amount of vasoconstrictor made, through hormones such as renin, ADH, aldosterone, AMP, and through the autonomic nervous system, given that more blood is sent to the kidneys under parasympathetic stimulation. So the first physiological thing that happens in nephrons and thus kidneys is filtration. The blood is filtered to remove urea. The second thing that must happen is that the good stuff must be reabsorbed, because as I had mentioned, that that while there may be 1 or 2 liters of urine produced in a day, there's 180 liters of filtrate. This filtrate includes glucose, amino acids, salts, things that we want to stay in our body. And so these other solutes, they must be reabsorbed. The key to reabsorption is sodium. All right, so understanding sodium is the key to everything else. There are sodium-potassium ion exchange pumps in the tubule cells. Now, when I say that, you're not surprised, because you're thinking, well, doesn't pretty much every cell have the sodium-potassium ion exchange pumps? Aren't these proteins among the most important in the body? And the answer is that's true. And so the pumps, which will take sodium, 3 ions at a time, from inside the cell and pump it out using ATP, and then take potassium ions 2 at a time from the exterior of the cell, here in blue, and pump them inside the cell. It is true that these proteins are pretty much in all cells, but in kidneys, they're special, because in the renal tubule, these pumps only exist on the side of the cell facing away from the kidney tubule's lumen and towards the blood vessel. So in other words, while this pump is active, these sodium ions are moving away from the filtrate, which is becoming urine, and towards the blood. In other words, the more this pump is used, the more sodium will be reabsorbed. Potassium, as we will see, the opposite is happening, because it's moving towards the tubule lumen. Before I go back to that video, just to repeat. So lots of cells have these sodium-potassium ion exchange pumps. But if you were to look in the tubule cells, they aren't here, all right, on the side of the cells which face the lumen of the kidney tubule. Notice the microvilli to help secrete and absorb things in the kidney tubule. Instead, the sodium-potassium exchange pumps are located here, the basolateral membrane, which face the blood vessels. So when these sodium-potassium exchange pumps pump sodium, for example, it's leaving the cell, going towards this blood vessel, so that it will be reabsorbed. So that's a key. These sodium-potassium exchange pumps are expressed in the basolateral membrane, not the apical membrane. When they are active, sodium then goes from these cells into the blood. That lowers sodium concentrations here, so more sodium comes in, it gets pumped out. More sodium comes in, it gets pumped out. More sodium comes in, it gets pumped out. All right, now, because of how much energy is spent to do this, A cell may spend 40% of its resting energy budget on sodium-potassium exchange pumps. It's not surprising now that there is this gradient of sodium going from the lumen into the blood. Now, sodium turns out being key to everything else, however, because when sodium goes from the lumen through the cells to here, chloride follows. Sodium is positive, chlorine is negative, and so the cell doesn't have to spend any energy. Chlorine just tends to follow the sodium gradient. Water will tend to follow, because if salt is moving this way, in osmosis, water moves to the solute. The solute's now going this way. So now water from the lumen is now ending up in the blood plasma. As I said, there's 180 liters of filtrate made in a day, but only 1 or 2 liters of urine. So most of this water ends up going this way. And this sodium gradient is key to that. And then plus there's more, because there are these special gates. Let's say that there's glucose in the lumen, and the body doesn't want glucose to end up as a waste and being flushed down the toilet. It wants glucose back in the cells and back in the blood. Well, here's a special gate where, in essence, the cell says, glucose, you want to come this way? You can, if sodium comes with you. See, the cell has spent so much energy to set up a sodium gradient. Sodium is going from an area of high concentration to low, that the cell can then take advantage of this gradient to move other items. So glucose will come into the cell if it moves with sodium, and thus glucose is reabsorbed from the tubule lumen. Amino acids, there are separate doors which allow amino acids to pass if if sodium comes with it. So the sodium gradient is taken advantage of again. These pumps are these Symport pumps. They are called Symport pumps because sodium and glucose both come into the cell together. They're moving in the same direction. So that's a Symport pump. They are in the microvilli at the apical portion of the cell. So sodium is key to all of these other substances moving. Many of these other substances are moving through secondary active transport. So the pumping of sodium was the primary active transport, which directly used ATP. But now that there's a sodium gradient, we can have a door where we say, amino acid, you can come into the cell if sodium comes with you. That's secondary active transport. And because they're both moving in the same direction, that is known as Symport. An enormous percentage of this reabsorption occurs in the proximal convoluted tubule. There are sodium pumps pumping sodium into the peritubular capillaries, and then chlorine follows, water follows, and then those Symport pumps then allow for the absorption of glucose, amino acids, and the like. And so along the PCT, about half of the water and salts are absorbed. All of the glucose should be absorbed, and all of the amino acids. And so a great deal of reabsorption occurs in the proximal convoluted tubule. Now, this is perhaps more a topic for lab, but if you were looking at what is abnormally found in urine, you might realize that glucose is tested for in urine. Why is that? Well, in a normal individual, while there is glucose in the blood, and thus glucose in the filtrate of the blood, the glucose is reabsorbed along the PCT. However, if one is diabetic, one's blood sugar can be elevated. And so now that means that there's an excess of glucose in the blood. That means there's an excess of glucose in the filtrate. While the pumps do reabsorb glucose, there is a limit to how fast they can work. There is a maximal amount of glucose which can be reabsorbed per hour. And because the glucose started off being so high in the plasma and then in the filtrate, there is glucose which isn't reabsorbed, and therefore it would be lost then in the urine, and that would be one of the possible tests for abnormal components of urine. So the PCT does a great deal of reabsorption. And the next section is known as the loop of Henle. The loop of Henle has two sections, a descending loop, which has squamous epithelial cells, and then an ascending loop, which has cuboidal epithelial cells. The descending loop can reabsorb water. The ascending loop can reabsorb salt. And by salt, I mean one ion of positive sodium, one of positive potassium, and two of negative chlorine. The reason that it is a loop is because each side now helps the transport in the other. When salt is pumped out of the ascending loop, this then draws water out of the descending loop, since water goes to where the salt is. So the salt, which is now outside the descending loop, helps to draw out the water. As water is taken out of the descending loop, that concentrates the salt. And this concentrated salt is now easy to pump out of the ascending loop. As more salt is pumped out of the ascending loop, that will draw more water out of the descending loop. As water comes out of the descending loop, this concentrates the salt and makes it easy to pump out of the ascending loop. And so the reason that the loop of Henle exists is that it is an adaptation for life on land, in terrestrial environments where water needs to be conserved, because each half of this loop of Henle helps transport something out of the opposite half. This is far more efficient than if it were a straight line, because these fluids are moving in opposite directions. This is an example of what is known as a countercurrent exchange. The reabsorption of the PCT in the loop of Henle is obligatory. It always happens no matter what. But once you get to the distal convoluted tubule and the collecting duct, reabsorption is possible, but it is influenced by hormones, and therefore it is what is known as facultative. It doesn't have to occur. So for example, if no hormone ADH is made, then the water which reaches the DCT simply continues and then will be lost as urine. And so not making the hormone ADH results in a dilute urine because that causes these water channels not to be synthesized, which would reabsorb the water. So hormones can cause a dilute urine to be made if ADH is not synthesized. If ADH is synthesized, then water channels are made, and this water is reabsorbed. And so urine can be more dilute than the plasma it came from, or it can be more concentrated based on which hormones are made. Aldosterone is a hormone released from the adrenal glands, which then does the same for the reabsorption of sodium in the DCT. So sodium reaches the distal convoluted tubule. If no aldosterone is made, then this sodium is not reabsorbed, and it will simply then continue and then be excreted in the urine. If, however, aldosterone is made, then the sodium-potassium exchange pumps along the tubule become more active, and the sodium will be reabsorbed. And so how much salt is lost in urine can be varied with hormones. Not making aldosterone will cause the release of excess salt, but making aldosterone will cause sodium to be conserved when it is in short supply. This is what's known as facultative reabsorption. So in the PCT and loop of Henle, there is obligatory reabsorption, which always occurs, while instead in the DCT and collecting duct, there is facultative reabsorption, which occurs based on which hormones are made and the physiological needs of the body. Okay. Finally, of the three, there are three functions of nephrons. Nephrons filter the blood to remove urea. They reabsorb the good stuff like water, salt, etc. And then finally, they can secrete additional waste. And so there is urea, which is in urine because it was originally in blood. But then there is additional waste that the tubule cells can release. Some of these wastes can travel because of the sodium gradient. So here's a door that says, sodium, the light pink, if you want to come in, you can, but only if something else leaves. And so hydrogen ions, they were the bright red, they were pumped out of the cell as sodium came in. And so this is also secondary active transport, but it is antiport because sodium comes in and potassium leaves. And so there can be more acid in the urine because of the tubule cells actively secreting an additional waste. Getting back to the sodium-potassium exchange pump, if this is the side of the cell where the blood vessels are, as we pump sodium in this direction, it is being reabsorbed. But in the DCT, when you have these pumps become more active, then the blue potassium is going this way, away from the blood capillary and towards the tubule lumen. So making the pumps of the DCT more active using aldosterone will cause more potassium to move towards the tubule lumen and thus be secreted. So while aldosterone is promoting the reabsorption of sodium because it's going this way into the blood capillaries where it will be reabsorbed and retained, potassium is moving this way towards the tubule lumen and thus likely to be excreted. And so there can be additional wastes which are secreted from the tubule cells. So there can be hydrogen ions, which are pumped out because of an antiport pump, ammonia ions, and then also potassium, thanks to the action of aldosterone. Now, this will come up in the next chapter as well, but the tubule cells make acid this way. They take carbon dioxide and water, put them together to form carbonic acid, and the carbonic acid then splits to make hydrogen ions, acid, and bicarbonate. The bicarbonate goes into the blood where it is a buffer, while the acid then can be pumped into the filtrate, which becomes urine. And so thus acid can be secreted from the body. Okay, and so urine can be much more acidic than the blood it came from because of these mechanisms. Just looking through the remainder of the playlist, if you needed a quick review of acids and bases, I have some resources here for you. And then there's a kind of a silly set of videos where all of the ions are talking at you with accents, and sodium ion talks to you and describes why it is leaving the body or entering where it is being reabsorbed and why. Potassium goes through this and why. And so if you wanted more review of that, those videos may be helpful. And then I have videos on urinalysis on what components shouldn't be present in urine and the conditions that would cause them to be in urine. So for example, glucose, bilirubin, high levels of ketones, blood, protein, nitrites, and leukocytes. These shouldn't be in urine, but various conditions like diabetes for glucose and ketone bodies, or bilirubin from liver damage, or blood, say from kidney stones, or damage to the filtration membrane. Proteins, the same thing, leukocytes or nitrites from urinary tract infections. There can be abnormal components in urine, and these videos then go through what is normally present in urine and what isn't. And so the physiology of the urinary system is rather simple. The kidneys are made of nephrons, and nephrons perform three functions. They filter urea out of the blood. They reabsorb good stuff at the renal tubules as opposed to the filtration, which occurred at the renal corpuscles. And they secrete additional waste like acid, potassium, and ammonia into the tubule. So that is the physiology of the urinary system.